Hello and welcome, dear students. How are you all? Okay, so your break's over now. So please, let's continue. Centers, I would ask you all to report your attendance, and we are beginning right away. Dear students, please get back onto your seats. Do that quickly. Okay. All right, so centers, please report your attendance, and we are beginning right away. Students, please report your attendance. Okay, thank you, Tamang Long students. Shillong. Okay, thank you, Shillong. Other centers, please report your attendance. I believe Churachandpur students are still remaining. Churachandpur students, please report your attendance. Please do that quickly. Okay. So in the meantime, first of all, we are beginning the revision, and then we'll get back to, in about 5-10 minutes, we'll get back to where we finished off last time. Right. So I was teaching you about photosynthesis, and photosynthesis basically means synthesizing food in the presence of light. The raw materials of photosynthesis are carbon dioxide, water, in the presence of sunlight and chlorophyll. They will make glucose or sugars or carbohydrates. Okay, general form is glucose, C6H12O6. And we make water also, and we make oxygen also. 3 products made from 2 raw materials. Yesterday we learned that oxygen in photosynthesis comes from the breakdown of water and not the carbon dioxide. Which experiment conclusively proved that oxygen in photosynthesis comes from water, not from carbon dioxide? Can you please answer quickly? Students, can you please answer that which experiment proved that oxygen in photosynthesis, the source is water? All right, so please do that quickly. You can take a look at your notebooks. There were 2 scientists. They used alga called chlorella, and they used oxygen-18 isotope. Rubin and Kamen. Okay, Rubin and Kamen proved that. And the first suggestive evidence came from Van Niel, but Rubin and Kamen basically proved it. Okay, yeah, thank you. You remember. And after Rubin and Kamen's experiment, there were some few experiments by other scientists, and they proved the existence of 2 phases in photosynthesis. One is called the light-dependent phase, the other is called the light-independent phase. Commonly we call them light reaction and the dark reaction. So today we are going to take a look at the details of light and dark reaction. Are you ready? All right, so let's get started from where we left off in the last class. So I was teaching you that in light reaction, we basically make 3 things: ATP, source of energy; reducing agent, which is NADPH2, and that's reducing power or reducing agent. It so provides compounds with the source of hydrogen, and hydrogen, when it is added, it becomes a reduction reaction. And oxygen is a waste product. It's a side product produced in photosynthesis, and it's not used in photosynthesis, but it is used in respiration. And rate of photosynthesis is many times that of respiration. That is why we say that plants breathe out oxygen during the daytime, but in the night time they breathe out carbon dioxide. Still, the concentration of oxygen in an atmosphere remains constant or increases or slightly decreases depending upon fossil fuel burning, etc. So why is that? Because of photosynthesis. All right. So I told you that light reaction basically means photophosphorylation, and photophosphorylation is of 2 types. The first type is called non-cyclic photophosphorylation or Z scheme. The other is called cyclic photophosphorylation. And we are going to take a look at these 2 types. All right, so let's get started with the Z scheme. And it has an interesting name. When I draw this Z scheme, you will see that it doesn't look actually like a Z, but more like an N. But it's just a manner of looking, okay? So let me teach non-cyclic photophosphorylation to you first. Non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Okay, you have a question? Sir, does photosynthesis still occur at night while the carbon dioxide is low? Yeah. I'll actually tell you the, okay, all right. It can occur during the night if you provide the plant with an artificial source of light, such as a CFL lamp, a tube light, or anything. It will not occur in moonlight because moonlight is not of the right intensity to cause photosynthesis. But yes, it can occur at night or at any time whenever you provide the plant with the source of light. When carbon dioxide is low, actually there is a thing called a compensation point. Look, whenever sun is rising, before that time, that period is called the dawn, and when the sun is setting, there is less sunlight, it is called dusk, okay? So the dawn and the dusk, basically the evening times and the morning times, during these times, the junction time of night and day, sunlight intensity is quite low. It gets increasing or it gets on decreasing. So there is something called a CO2 compensation point, and at these points, the amount of photosynthesis, somehow what happens is, the amount of oxygen released is equal to carbon dioxide consumed or released, sorry. Oxygen released is equal to amount of CO2 released. So for C3 plants, this compensation point comes at around 25 to 100 ppm when CO2 is equal to oxygen. For C4 plants, it is very low. It's about 5 to 10 ppm. All right, parts per million. ppm means parts per million. The normal concentration of oxygen in the atmosphere, whether it is day, whether it is night, it is 360 ppm. Okay, or 0.036%. Okay, so normally the oxygen concentration at night is not different during the day. It is not different during the day, it is not different during the night. Okay? So it's always going to be 360 ppm. However, if you measure the CO2 concentration in a closed atmosphere, closed region, like you close some people together or sitting in a classroom, and the classroom's doors are locked, windows are locked, there is very less source of ventilation. Under that condition, you can say the concentration of CO2 will not remain 360 ppm. It will rise to about 400 ppm or 500 ppm. Okay, you can judge that this is happening when everybody is yawning. When everybody is yawning, then that means you are trying to open your mouth and take a lot of oxygen inside, so that basically your muscles can start working again. They can lose the extra carbon dioxide quickly. Okay, yawning refers to when the CO2 concentration is in your body is great, right? So whether it is day, whether it is night, it's always going to be 360 ppm in open atmosphere, right? All right. So let's continue. Non-cyclic photophosphorylation, or the other name is Z scheme. So what happens is, non-cyclic photophosphorylation, I'm going to draw it in a next page because it requires a lot of space to draw it. We will begin this reaction with photosystem 2, PS2 or photosystem 2. Photosystem 2 has chlorophyll A 680 nanometer as the reaction center. On absorbing sunlight, its electrons get excited, and these electrons, how much are released at a single point of time? 2 electrons will be released. 2 electrons will be released from photosystem 2. Okay? Each electron, they will be basically accepted by an electron acceptor. So I'm writing 2 electrons here, and instead of inside writing 2 electrons, I will write electron acceptor. Primary electron acceptor. Electrons get excited, accepted by the primary electron acceptor. Now please try to understand why is this happening. This process is similar to when you are taking something, lifting it up and then dropping it down. Okay? Why has this page of paper the tendency to fall down? Why is this page of paper have the tendency to fall down? Because when I lift it up, it gains some potential energy. Similarly, when the electrons get excited, they have a lot of potential energy, and this potential energy can be used to capture energy and make ATP out of it. Are you understanding me? Electrons getting elevated to a higher position means they have more energy. Okay? So, more energy electrons, when they will come down, I will capture these electrons, use that energy to make some ATP out of it. It is just similar to a hydroelectric dam. Water is stored at a higher point. It is released from there. It is used to turn a turbine, and in here there is a turbine also. Do you know the name of the turbine that is existing in both photosynthesis and respiration? Do you know the name of the turbine? Anybody? Similarly, in ATP synthesis, we have a turbine called ATPase. ATPase molecule is in actuality like a huge molecule. It's like a turbine. If you put adenosine diphosphate and inorganic phosphate into this turbine, it will turn and it will cause formation of ATP by joining ADP and IP phosphate, and ATP will be produced. So just like the turbine in a hydroelectric dam produces energy, here this turbine of ATPase enzyme will produce ATP. All right, moving on. This is photosystem 2 on getting photons from sunlight. It gets excited. These electrons are accepted by an electron transporter or acceptor, and these will play catch and throw. Okay, so some people of you can make some paper balls, and I see some people of you taking that paper ball and passing it to someone else, and someone else passes it to someone else. So this game keeps on going on. So the students who play these games, you can compare them to electron acceptor or pigments called cytochromes. Okay, I know the students particularly in Shillong. I almost recognize your faces also. So who play all the time. At least during the break I can see you. So yeah, so you people are equal to cytochromes. And cytochromes, what do? They take the electrons from one electron acceptor, pass it to the one behind them, and the behind them electron acceptor passes it to someone else. And during this time, when there is a sufficient energy gathered, at one point of time when we have, look, the ball actually, the electron actually, when it is coming down from the primary electron acceptor, this ball is very, very fast. This electron is very, very fast. You cannot capture this energy. You can only capture this energy when it has reached an intermediate speed. When it has reached somewhat intermediate speed, so that your enzyme does not get damaged and you have sufficient energy also. So we cannot capture it, this energy, when it is very fast. We cannot capture this energy when it is very slow. So somewhere in between there comes a perfect balance. This balance comes in the form of cytochrome B6, cytochrome F. In between there are other electron acceptors, plastoquinone, plastocyanin, pheophytin. But we are not going to write these names because these are not in your course. I'm going to focus simply on what's given in your NCERT right now. If you want the more details, I can give you another diagram, but that time will come later. So primary electron acceptor accepts the electron and passes it to another electron acceptor. This electron acceptor passes it, takes it, and then releases it. So basically it is passing again. And here comes a complex called cytochrome B6 F complex, a combination of two cytochromes. If you want to write them together, you can write cytochrome B6 F complex. Okay, and during this time when cytochrome B6F complex is working, here is the time when I use ADP plus inorganic phosphate to make ATP. All right. This electron is transferred to another electron acceptor. All right. And finally, these electrons are given to photosystem 1, which has lost all its electrons, sorry, not all its electrons, which has lost 2 electrons by sunlight falling on it. Photosystem 1 absorbs light of even longer wavelength. So that is why I have drawn photosystem 2 with orange color, this with red color. You do not need to write, draw with red and orange color. It's just a representation, okay? So I'm writing just sunlight here. And because of losing the electrons, it has become very deficient, and these electrons deficiency is completed by the electrons coming down, and these electrons are given by electron acceptor. So its deficiency will be fulfilled. The electrons given out by photosystem 1 are accepted by another electron acceptor, okay, taken by another electron acceptor, and this electron acceptor takes these electrons and passes it to an enzyme called NADP reductase. It is given to NADP reductase. NADP reductase. I hope it is visible to everybody, right? NADP reductase. If it's not clear, I'll write it again with larger font size. An enzyme is present here, NADP reductase. The NADP reductase gives these electrons in turn to NADP plus, and we get NADP plus accepts the electrons from the electron acceptor. Sorry, I should draw it like this. Yeah. And another thing, 2 protons are here. I will draw this proton with a particular color because it is very important that you remember where these protons are coming from. And then we get NADP H plus H plus, or you can write it as NADP H2. There is a bit of chemistry involved here. There is no need for people to get worried. Don't be afraid of this chemistry. I'm going to explain everything again and again till you totally understand what is happening here. There are a lot of things that are happening here. I'll explain them again till you people understand it totally well. Okay, so no need to get worried. There are a lot of complexities here. Don't get worried. It's a bit difficult to learn this, but I'm sure you will be able to do it. Now you have drawn this, and now I can try to finish some fill in the blanks that are left incomplete here. There are some things I need to fill more than this diagram that is given here. Okay. Whenever you're ready, let me know when you have drawn it, because there's some things that I need to draw more in this diagram. But I can do that once you finish. So let me know when you're finished. Okay. Coming along, you have done. Shillong students are still drawing, I believe. Shillong students, please finish fast. I'm waiting for you. Please draw everything in the correct sequence. I would like that you draw it in one full page. At least I hope that you must have drawn it in half page. Do not draw it very short, because if you draw it very short, things will not be very clear. Okay. Shillong, are you done? Okay. So let me begin, because I believe most of the people have almost finished this diagram now. All right. So photosystem 2, it has a reaction center which is chlorophyll A, with the wavelength peak absorbed at 680 nanometer. Photosystem 1 has a chlorophyll which has a peak absorption wavelength at 700 nanometer. All right. Okay. So what happens is, chlorophyll molecule is having a tetrapyrrole structure in which magnesium ion is sitting in the center, and when it gets sunlight, these electrons of the electron cloud or the electron rich region of surrounding the magnesium ion, it gets very excited. Okay? Suppose a guest comes to your home and they give a 2,000 rupee note to you. Will every one of you be excited? Yes or no? Yeah. And what happens is, you get a 2,000 rupee note from the guests that have come, and whenever they are going away, they give a 2,000 rupee note to you. So you will be excited, hyper excited, I must say. So you will be so excited that you will leave your home and go anywhere else for shopping or anything. Okay. So you find someone there, and your friends are looking for the money that you haven't paid them since 1 or 2 years. So you were the primary electron acceptor, but now your energy has decreased somewhat because somebody else is taking some money from you. And finally, there comes someone else, and your mother sees you shopping, and she takes whatever money is left from you and gives you only 100 rupees or 50 rupees. So your mother or father, you can compare them to cytochrome B6F complex, which are taking your money or your energy and getting it converted into something useful, and whatever energy remains, you invest it into a friend that you believe will come of use in some time later. Okay. So that friend is photosystem 1, and this friend also gets excited from time to time, and when it gets excited, it also invests its money in something useful, and this something useful, another something useful is called NADPH2, or reducing power. So cutting out all that talk, let me focus on the chemistry part now. This was just a story to help you understand what is happening here. Photosystem 2 gets sunlight, absorbs photons of sunlight, and gets excited. When it gets excited, it loses its electrons. When it loses its electrons, whenever any species for that matter loses its electrons, it becomes an oxidizing agent. Are you getting me? If something is electron deficient, it represents oxidizing agent species. Everybody of you knows oxidizing agents. Can you name some oxidizing agents other than what are shown in this diagram? Can you give me examples of some good oxidizing agents that you use in your lab? K2Cr2O7, KMnO4, right? These are 2 oxidizing agents that you use. Besides this, sulfuric acid, nitric acid, all these are oxidizing agents. An oxidizing agent is a species which has some central element or metal which is in high oxidation state. KMnO4 has manganese in +7 oxidation state. When it is mixed with something, it oxidizes other things and reduces itself. It gets reduced. Its oxidation state becomes less positive, right? So this is happening. Similarly here, chlorophyll A680, when it loses the electrons, it becomes a powerful oxidizing agent. It becomes so powerful that it can oxidize or break down water. Are you getting me? PS2, when it loses the electrons, it becomes highly unstable and it becomes a very good oxidizing agent. It needs electrons. It has been snatched of its electrons and because it has been snatched of its electrons, it will take them from someone else who can give the electrons. And this species is water. I'm drawing it with blue color. Water molecule is broken down by photosystem 2. And when photosystem 2 takes the electrons from water, it gets reduced. Photosystem 2 oxidized plus water gives photosystem 2 reduced plus 2 protons plus half oxygen. I'm going to change the color of the protons here so that you can remember something. Remember the protons here, right? These are the protons. These are the protons of NADPH2 which are coming from the breakdown of water. And where have the electrons gone? Electrons have gone to photosystem 2. Okay. So the 2 electrons of oxygen have been taken by photosystem 2. All right. So this is clear to everybody. So charge, you can say that here photosystem 2 was in +2 oxidation state, and when it has been reduced, you can say it has changed to maybe 0 oxidation state, but that is not accurate because we cannot locate the exact source of deficiency. Anyway, there is a very important part that I want you to remember. Can you name 2 mineral ions that are essential for photolysis of water? I have told you the answer when I was teaching you mineral nutrition chapter. Anyone who answers it correctly, they might get a chocolate from me whenever I visit you. Yeah. They get at least a dairy milk. Yeah, it's not a very hard question. So I'll give you a dairy milk for this. Whenever I visit you or you come here. All right. So this question is not very hard. I told you the answer when I taught you mineral nutrition. Can you remember the name of these 2 mineral ions? Please do that quickly if you can. Nobody? I taught you the answer, and stressed this to you that it is asked in the NEET examination. What are the 2 mineral ions that are essential for photolysis of water? Breakdown of water, it is also called photolysis of water. I have told you the answer when I was teaching you mineral nutrition. It is not given in your NCERT in photosynthesis. The answer is given in NCERT in mineral nutrition chapter. Yeah, somebody is typing. I think you can answer now. Somebody got the answer, I think. 2 mineral ions. Come on. Time is up now. It's getting really late. All right, I'll write them for you. Manganese and chloride ion. These 2 ions are essential for the breakdown of water. If you do not add these ions, if these 2 ions are absent, photolysis of water will not happen. Okay, nobody answered. Okay, no problem. I'll wait for you to be able to get better and answer me quickly. That day might soon come. Photosystem 2 oxidized. reduced by the electrons of oxygen ion. Okay, oxygen is in -2 oxidation state. On the right hand side, it is in 0 oxidation state. So basically, oxidizing agent gets reduced like manganese, KMnO4. KMnO4, you react it with something, it gets oxidized, it gets reduced into +2 oxidation state. All right. So I'm just telling you all this. It's a part of chemistry. No problem. Okay. So basically, we have learned this, that when photosystem 2 loses the electrons, then it gets its electrons back by breaking down water. Now, photosystem 2 electrons are taken by the primary electron acceptor. This means that the electrons lost by photosystem 2 are never going to come back to itself. These electrons are taken by one electron acceptor, passed to another, just like playing basketball with it, and then finally it is given to photosystem 1. Photosystem 1 also lost its electrons, and these electrons that were lost are basically they are made up for by electrons coming from PS2. The PS1 electrons are taken up by another electron acceptor. This electron acceptor takes 2 protons or H+ ions formed by the photolysis of water and NADP+, and these 2 electrons are given to NADP reductase. So net result we get is NADPH2 or NADPH plus H+. There is hydrogen inside this molecule. Everybody can see hydrogen inside this molecule, right? Hydrogen inside this molecule. This is the reducing power. So now see what are you getting from non-cyclic photophosphorylation. You're getting 3 things. You're getting ATP, you're getting NADPH2, and you are getting oxygen. If anybody is left out in writing some details, please do that quickly. The 3 products of photosynthesis, reducing power, energy source, and oxygen, have all been made now. So let's write this down now. I believe everyone has completed the diagram and everything. So let's continue. What is happening in non-cyclic photophosphorylation? Please write. In non-cyclic photophosphorylation, the events start with PS2 or photosystem 2 absorbing sunlight and getting excited. Getting excited, just like getting a 2000 rupee note or even 2000 less. So let's make it more interesting. Someone gives you $100. $100 must excite you because that represents a significant sum of money. You can convert it into Indian rupees and say where you are getting at. So the events start with PS2 absorbing sunlight and getting excited. PS2 on getting excited loses 2 electrons. loses 2 electrons. The electrons which are lost, okay, the lost electrons are accepted by primary electron acceptor. Primary electron acceptor. On the other hand, PS2 without electrons becomes a very strong oxidizing agent. Becomes a very strong oxidizing agent. And this strong oxidizing agent, I'm going to mark it with a star. When it is excited, I'm going to mark it with a star. PS2 oxidizes water molecule or breaks down water molecule. So you can say PS2 which was excited plus H2O gives PS2 reduced plus 2 protons plus half oxygen. This reaction is called photolysis of water. This reaction is called photolysis of water. Okay. You have done? All right. This reaction is called photolysis of water. And photolysis of water is an important step and requires presence of manganese and chloride ion. Okay. So you can write here Mn plus 2 and Cl minus are essential for photolysis of water. Essential for breakdown or photolysis of water. All right. Okay. Now we can continue with the Z scheme. Now we can write about the rest of the reactions. The rest of the reactions that are happening is the primary electron acceptor now loses electrons and passes them to another electron acceptor Okay. The electrons represent potential energy and this energy is used in making ATP. So when electrons are being passed from cytochrome B6 to cytochrome F, then there is an ATPase enzyme which uses energy to make ATP molecule. All right. And what's more, then cytochrome B6 passes to cytochrome F, and the electrons that are lost by cytochrome F passed on to another electron acceptor. Finally, after passing through many electron acceptors, electrons are given to the photosystem 1, which is an oxidized state. After passing through other electron acceptors, after cytochrome F, these electrons are finally given to oxidized photosystem 1, which becomes reduced or normal now. When it is normal, means it is again ready to absorb new photons. The electrons released by PS1 are given to another electron acceptor, another electron acceptor, which passes it to reductase enzyme and NADP plus. NADP reductase. Now finally we have NADPH2. Or you can also call it NADPH plus H1. Sorry, NADPH plus H plus. It's a manner of representation. If I don't write it down, everybody will get confused. NADPH plus H plus. One H is representing, it is coupled, other represents charge. It does not mean it has +2 charge, okay? So that might become confusing if you don't know how it is written. So finally, we have made 3 things in Z scheme. Therefore, the products of non-cyclic photophosphorylation or Z scheme are can you write it down by yourself? What are the 3 products? Should I write them again? Yeah. Reducing power, NADPH2. Second is energy, ATP. Third is oxygen. So I believe you must have finished now. So why it is called Z scheme? Let's take a look. Why it is called Z scheme? It's called Z scheme because the path of movement of electron looks like Z. It looks like Z. You can also say that it looks like N. But considering how far the electron is going, so it is closer to Z other than N. Okay. You can ask the scientists who named it as Z scheme and not N scheme. Okay. So maybe I would have named it N scheme, but the scientists decided to call it Z scheme. Okay. So this is the reason why we call it as Z scheme. Coming back, is there anything left now? I hope you have finished writing. reducing power, ATP. These are the essential things, and oxygen is just a waste product. So everybody must have done it by now. Okay. So we finish non-cyclic photophosphorylation. There is a last thing remaining here, Hill's reaction. Hill's reaction. Light reaction is also sometimes called Hill's reaction. Why we call it as Hill's reaction? There was a scientist called Robert Hill. Robert Hill took some isolated chloroplasts and treated them with electron acceptor. Electron acceptor. What do you think will happen? What do you suppose will happen? Any idea? Okay. Basically what we are doing is, we are taking some chlorophyll and treating it with electron acceptor. The electron acceptor that is taken is iron oxalate, iron in +3 oxidation state. So it will accept electron from chlorophyll, and what will happen then? Because this medium is aquatic, in aqueous medium, so this means that if I take chlorophyll from isolated chloroplasts and treat it with iron +3, and it is done in presence of water, what will I get? I will get iron in reduced state, +3 to +2, and chlorophyll, basically what does it do? Chlorophyll breaks down. Chlorophyll actually, this is an electron acceptor. Iron +3 takes electron from chlorophyll and converts into iron +2. And chlorophyll takes electrons from water. So chlorophyll is also reduced, first oxidized, but then reduced. And what will happen to water? Water will become oxidized, and you will get 2H plus half oxygen. Now this reaction is balanced. Now this shows that oxygen is released. So basically chlorophyll is doing something. When he performed this reaction, he saw that oxygen was being evolved. Oxygen can be evolved only when iron takes electrons from chlorophyll and chlorophyll takes electrons from water. So basically oxygen evolution, he proved that oxygen evolution takes place in light reaction. Takes place in light reaction. So this is also called Hill's reaction. Now continuing, cyclic photophosphorylation. I hope you do not need a break right now, okay? So if you need any such thing, let me know later, because I won't be able to give a longer break, maybe 1-2 minutes. That's all today. I want to finish a lot of things today. All right. Second part is cyclic photophosphorylation. Cyclic photophosphorylation. In cyclic photophosphorylation, what happens is, in cyclic process, we take the electrons and give it back. Take the electrons, give it back to the source where it originated. Here, the only photosystem that is involved is photosystem 1. Photosystem 1. It takes electrons of sunlight gets hyperexcited, gives it to electron acceptor, electron acceptor takes the electrons, uses it to make ATP and passes it back to photosystem 1. So you are only making one thing here, and that is ATP. That is ATP. So the only product of cyclic photophosphorylation, it is very easy. Only non-cyclic is a bit tough. Okay. So you can write now here. The product of, only product of cyclic photophosphorylation is ATP. Okay. So this is about cyclic and non-cyclic photophosphorylation. With this, I can say that light reaction is finished. Light reaction is done. Can you also remind me where light reaction takes place? It takes place in the grain of chloroplast. Grain of chloroplast. So you are finished with the light reaction, and the thing is, you are making ATP in one process and ATP, NADPH2 and oxygen in the other process. Now all these things, these are the raw materials for which we can begin the dark reaction. So now please put the heading dark reaction. dark reaction. Wait a second. This is not cool. Yeah, now it might look a bit cooler. Okay, now I can begin what I wanted to do. Dark reaction. I wanted to display face to look like more like Joker's face. Okay, just wanted to add some things to it. Anyway, so now we can begin the dark reaction. Alright, so what does dark reaction refer to? You can say dark reaction refers to dark reaction refers to the light independent phase in which materials manufactured during the light reaction phase are utilized. So we are going to basically utilize what we had made in the light reaction, right? So let's continue. Its site of dark reaction, we must also complete this first. Site of dark reaction. Stroma of chloroplast. Stroma of chloroplast. Okay. So the basic site of dark reaction is stroma of chloroplast. So let's continue that. Stroma. Continuing, we have to tell you now the place where dark reaction is happening. Okay, it's an overlapping issue now. Let me do it again. Just write it simply. It's this stroma. And it will write again later. I'm sure of it. It's not appearing. Okay, anyway. Now you know that it is this stroma. So let me continue with how it is happening. The dark reaction was discovered by the reactions of dark phase were discovered by Melvin Kelvin. Were discovered by Melvin Kelvin. This Kelvin and the scientist who proposed the Kelvin scale, they are both totally different. Kelvin was a chemist, and the Kelvin of Kelvin scale, who gave the formula of converting Celsius and Fahrenheit into Kelvin, that was a physicist. This Kelvin is not even a biologist, he is a chemist, and he gave the dark reaction. So dark reaction is basically divided into three phases. Dark reaction is basically divided into three phases. It is basically divided into three phases, and I must draw them. The first phase is the acceptance of carbon dioxide by something called ribulose 1,5 biphosphate. Ribulose 1,5 biphosphate is written as RUBP. Okay, and RUBP is going to accept a molecule of carbon dioxide. Okay, and it is going to accept the carbon dioxide. So basically this is the entry point of carbon dioxide. Carbon dioxide plus RUBP. Okay, and carbon dioxide is a 1 carbon compound. This is 1 carbon compound. RUBP is a 5 carbon compound. Okay, so 5 plus 1 gives 6. So when you combine them together, you will get a 3 carbon compound, 2 molecules. Actually, first of all, when it combines, it gives a 6 carbon compound, but 6 carbon compound is very, very unstable. And because of the instability, we are not going to discuss it here. And this is what is happening now. 6 carbon compound gets decomposed into 2 molecules of 3 carbon compound called 3-phosphoglyceric acid, 3-PGA. Okay, and 2 molecules are being synthesized. You can write 2 into 3-PGA. this enzyme that is involved in the carboxylation step just now, it is called Rubisco. Rubisco. RUBP is, I will write all these things here. First of all, just write this. This step here, it is called carboxylation. Is called carboxylation. Yeah, I rubbed a lot of part, I think. Okay. Yeah, well, rubbing in this is so difficult, so I'll just make it whole colored so that otherwise it does not get obstructing in my way. Otherwise, there is a huge problem here. So let me hold this whole black, and then I can write whatever I want to here. Just give me 5 seconds. Okay, now I can do what I want to. Writing the dark reaction in dark phase is much better. Now it's black as night. I hope you all like it. CO2 plus RUBP. RUBP is for ribulose 1,5 biphosphate. Gets converted into 3 carbon compound called 3-phosphoglyceric acid, 3-PGA, and it is having 2 molecules here. 2 molecules of 3-phosphoglyceric acid. CO2 and RUBP. RUBP is a 5-carbon compound. CO2 is a 1-carbon compound. This reaction is taking place with the help of Rubisco enzyme. Rubisco is the name of enzyme which is making this possible, and 3-PGA, formation of 3-PGA is called carboxylation. Carboxylation, right? And 3-PGA gets converted into something else by the help of ATP and NADPH2. NADPH2 will get converted into NADP+. So this step is called reduction. Reduction phase, carboxylation phase, and 3-PGA gets converted into something called triose phosphate. It is a 3-carbon compound. Actually, the chemical formula is 3-phosphoglyceraldehyde. Triose phosphate. Right? Triose phosphate. And this triose phosphate, it's a 3-carbon compound, and it has 2 molecules again. Okay, 2 molecules again. From these 2 molecules, I will again make RUBP, which is a 5-carbon compound. I will again make RUBP, which is a 5-carbon compound. So this next step, I will call it regeneration. Regeneration. Regeneration step, I will make RUBP again. So let us see what is happening. RUBP is a 5-carbon compound. From that you made 6-carbon compound, or basically you made something out of 6 carbons. Carbon dioxide is a 1-carbon compound, this is clear. 3-PGA is a 3-carbon compound. Okay. But when you make triose phosphate, and it is also a 3-carbon compound, from that you are making something which is 5-carbon compound. So there is loss of 1 carbon here, right? Where does this carbon go? Where does this carbon exactly go? So this carbon is taken out from here, and this is the reduced carbon. This represents the reduced carbon. This represents the reduced carbon. This represents the reduced carbon molecule, and this reduced carbon molecule, I am going to take it and I am going to form CHOH from it. Okay. So one more thing here. So this is basically reduced carbon. In one cycle, let's say I will make 1 reduced carbon, CHOH suppose. Now this reaction, let me summarize it. From 5-carbon compound, you have made a 3-carbon compound. I'm going to write all their names here. RUBP is ribulose 1,5 biphosphate. Okay. PGA is phosphoglyceric acid, or phosphoglyceric acid gives an ion, so it becomes glycerate. Triose phosphate is basically 3-phosphoglyceraldehyde. Whenever acid is reduced, you must have learned it in aldehyde and ketone chapter, it becomes aldehyde. So there are 3 steps here. First step is carboxylation. The second step is reduction. Third step is regeneration. There are 3 steps in this cycle called Calvin cycle, discovered by Melvin Calvin. And please see here, 3-phosphoglyceric acid is a 3-carbon compound. So this cycle is also called C3 cycle or Calvin cycle. In some plants, there is a 4-carbon compound formed, so we call them C4 plants. Most common plants are C3 or C4. C3 plants are most common. Now I am going to complete this Calvin cycle. See carefully. I am taking 1 carbon dioxide and I am giving out 1 reduced carbon in one cycle. So if I run this cycle 6 times, if I run this cycle 6 times, 6 times, right? Then I will get 6 reduced carbon, and this 6 reduced carbon is equivalent to, or it is equal to C6H12O6 glucose or fructose. Combine glucose and fructose make sucrose. This is the complete Calvin cycle. Actually, it is still not complete. If you want the complete Calvin cycle, I will have to give you 30 chemical reactions that are totally happening in the Calvin cycle. But you must be very happy that you should be happy, because some years back this Calvin cycle was very complicated for most students because 30 chemical reactions are not easy to remember. Just imagine if you had 30 chemical reactions. Would you be able to do it still in that case also? Would you be able to do it? It would have been very difficult. Okay. So this is the complete Calvin cycle. First step in Calvin cycle is carboxylation. The second step in Calvin cycle is reduction. The third step in Calvin cycle is regeneration. Everybody is finished? Should I move on to the next page? Please let me know if you are finished. Let me know if you are finished, students. Okay. Done? I'm wondering if why is it so long? Why not replying? So I'll move on to the next page. Calvin cycle is divided into 3 phases. Consists of following steps. Following main steps. First is carboxylation. In carboxylation step, RuBP is a 5-carbon compound, accepts 1 CO2 molecule and gets converted into a 6-carbon compound which is unstable, and it gets decomposed into 2 molecules of 3-carbon compound called 3-phosphoglycerate. Okay. This reaction takes place with the help of an enzyme called Rubisco. Rubisco enzyme, the full name of Rubisco enzyme is RuBP carboxylase oxygenase enzyme. But since it is doing carboxylation activity here, so I'm going to remove the word oxygenase and I'm just going to call it as carboxylase. The thing about Rubisco is, Rubisco is the most abundant enzyme in the whole world, or most abundant protein in the whole world. Almost 50% of the world's total protein is Rubisco. 50%, that's a huge quantity of all proteins, is the most abundant protein or enzyme in the world. So this is carboxylation. Second step is reduction. Reduction means use of reducing power. Reducing power is nothing else than NADPH2. So reduction step involves reduction of 3-PGA into 3-PGAL or triose phosphate. Actually, it is also converted into 3-dihydroxyacetone phosphate, but that is not required here. 3-phosphoglyceric acid is converted by ATP and NADPH2 and NADP plus comes out and ADP comes out and inorganic phosphate comes out. This energy and everything is used to make triose phosphate. I'm just going to write triose phosphate here, otherwise you might get confused. Reduction. Enzyme involved is reductase. Third step is regeneration. If you do not perform regeneration, this cycle will end here. It will not remain a cycle. If you want the cycle to begin again, you must make the compound which has been lost. So RUBP has been lost, I will make RUBP again. How will you make it? Convert 2 molecules of triose phosphate into RUBP plus 1 reduced carbon compound. This is regeneration. These are the 3 steps of Kelvin cycle. Is there anything you would like to know? Is there any confusion in this regard? If you repeat this cycle 6 times, you will get 6 reduced carbons. So for making 1 molecule glucose, we have to run the Kelvin cycle 6 times. So now write the last part. To make 1 glucose molecule, we need to run Kelvin cycle 6 times. Run Kelvin cycle 6 times. All right. And there is one more very, very important statement. What I want you to tell now is, to make 1 molecule of glucose, you need 18 molecules of ATP and 12 molecules of NADPH2. To make 1 glucose, what is the cost? You need to make, please write it and learn it by heart. Yeah, you can learn it, because if you want to go into the biochemistry, that normally people do when they do graduation at the IITs or in the NITs or in the Indian Institute of Science. So no need to run after the biochemistry or the BSc part. Just let's people who do biotech and take other subjects, they need to understand this in detail. You just need to learn this statement. Please write, cost of making 1 molecule glucose is 18 ATP plus 12 NADPH2. If you want to make 1 mole of glucose, then you need 18 moles of glucose and 12 moles of NADPH2. In C3 plants, in C4 plants, this cost becomes higher. You need 30 ATP, 12 NADPH2. I will explain later how that happens. And you are running the Kelvin cycle 6 times. So can you tell me how many molecules of how many molecules of ATP and NADPH2 consumed in running 1 Kelvin cycle? In 1 C3 cycle, how many ATP, how many NADPH2 will be used? Can you calculate? To make 1 glucose, you need to run cycle 6 times. And to make 1 glucose, you need total 18 ATP, 12 NADPH2. So what is the cost of running Kelvin cycle? Calculate and let me know. And please do that quickly. Cost of running one Kelvin cycle is 18 by 6 and 12 by 6. 3 ATP, 2 NADPH2. ATP, therefore, ATP and NADPH2 consumed in running 1 Kelvin cycle, 1 C3 cycle equal to 18 by 3 ATP plus, 18 by 6 ATP and 12 by 6 NADPH2. This gives us 2 ATP and 3 ATP and 2 NADPH2. So irritating. 3 ATP and 2 NADPH2. Now light reaction is finished, dark reaction is finished. Small topics remain: Blackman's law of limiting factors, CAM cycle and C4 cycle. That is the only thing remaining in photosynthesis chapter now. So time is a bit short. We will finish it in the next class. So please finish this quickly. Write whatever you want to, and we'll meet in the next class. Okay. So that's all for today, dear students. Thank you for your time and your patience. And we'll meet in the next class. We'll finish photosynthesis, and then we'll start respiration. Thank you.